Good morning, class. Today in this lecture talk, I'm discussing with you animal biotechnology uh, paper. Under that, I'll be dealing today culture media. In that, I'll be dealing today natural media. Okay. Animal cell culture can be described as an in vitro technique of maintenance and propagation of animal cells using a suitable nutrient media. Okay, in animal tissue culture or animal biotechnology, the most important thing here is that nutrient media. Okay, growing tissues in vitro is made possible in an appropriate culture medium containing mixture of nutrient either in solid or liquid form. Okay, what is this culture medium? Culture medium is a nutrient preparation which provides optimum condition for the growth and proliferation of cells. Okay, are microbes, a plant or animal cells in vitro condition. Okay, by maintaining the pH, optimum pH and osmotic pressure, etc. of the cells. Choice of media for the culture of animal cells mainly depends on type of cells to be cultured. Okay, that may be a normal cells, or we can also use immortalized cells. Normal cells requires defined quantities of proteins, growth factor and hormones. Okay, whereas immortalized cells, which require supplementation of external growth factors. And the next one is transformed cells. Okay, here the extra supplementation of growth factor as well as certain transformation factors such as transferrin we need to provide. Okay, so you can keep in your mind here normal cells and immortalized cells. Okay, then about this transformed cells. Okay. Next, let us go through here basic requirements for culture medium. Okay, to culture animal cells, all type of media must be composed of many essential components. Okay, which are used by cells for their growth, proliferation, and to differentiate during culture. Okay, in that some of the basic chemical requirements, carbohydrates, which acts as a natural energy source, which are used in the form of glucose. Okay, vitamins, essential for cell proliferation and differentiation and cannot be synthesized by cells during culture. Okay, inorganic salts which help to retain osmotic balance of cells. For this, we are using here potassium and calcium ions in media. Okay, amino acids include for proteins synthesis by cells because cells unable to synthesize them mostly, that is L-glutamine are used. Further, antibiotics are used to avoid to culture from microbial contaminants, okay, like viruses and other fungal, bacterial, etc. Penicillin and streptomycin, which is most commonly used as antibiotics. Next, trace elements for serum-free media in this include copper, then selenium, zinc, etc. Okay. Further, the requirement buffers which control pH. Okay. HEPS 42, that is 242 hydroxy ethyl 1 piperazine ethane sulfonic acid. Okay. Here H stands for hydroxy. Then this is ethyl. Then P, that is piperazine. Okay. Then ethane, and this S stands sulfonic acid. Okay, these are some of the basic requirements for culture medium. Let us understand again further about culture media. So what is this culture media? It is a mixture of nutrients required for the survival as well as growth of animal cells. Okay, and this must provide nutritional, hormonal, and stromal factors. Okay, based on the constituents of the media, there are two types of media, that is natural and artificial medium. Depending on the type of cells to be cultured, normal, immortalized, or transformed cells, okay, we need to think about the what kind of chemical nutrients we require. Okay, and here the objective of culture is to grow or their growth, then survival, then differentiation of the cells, then production of desired proteins. Okay, then by keeping all these things in mind, the culture medium will be selected or chosen. Okay, now let us go through here classification of culture media. We have classified this animal cell culture media into natural media and artificial or semi-synthetic media. Okay, so under natural media, you can recall clots. Okay, the next one is biological fluids. The next one is tissue extracts. Okay, this come all these which comes under natural media. Don't forget, kindly make a note. Next culture medium is artificial or semi-synthetic media. In that, we can recall serum containing media, serum-free media, chemically defined media, protein-free media. Okay, all this which comes under the culture medium. Hope you are able to understand. Now, let us first go through natural media. Natural media consists solely of naturally occurring biological fluids. Okay, natural media are very useful and convenient for a wide range of animal cell culture. The major disadvantage of natural media is its poor reproducibility due to the lack of knowledge of the exact composition of these natural media. As the name suggests, natural media, we won't be able to understand the proper chemical composition in that particular natural media. That was the only drawback or disadvantage. Okay, they may be in the form of fluid or maybe extract and coagulant or maybe a clots. Okay, this natural media can also be known as undefined media. We won't be able to define definite amount of the chemicals which are present in the natural medium. So we can also say this natural media as undefined media. Okay, it consists of the materials which are solely of natural origin. Okay, and here exact quality and quantity of the components are not known and cannot be determined exactly. Okay, but this natural media is mainly used as natural components. Okay, as clots or maybe a plasma, a clots or maybe a biological fluids and tissue extracts. So you can keep in your mind what are the natural media or components which are used in animal tissue culture, that is clots or plasma clots, biological fluids and tissue extracts are some of the examples for the natural media. Okay, where we are using all these components. Now let us understand first coagulant form or clot. Clots, the most commonly used clots are plasma clots. Plasma is commercially available in the form of liquid plasma kept in silicon ampoules or lyophilized plasma. Okay, plasma can also be prepared in the laboratory, taking out blood from male fowl and adding heparin to prevent blood coagulation. Heparin, which is going to prevent the blood clotting or blood coagulation. Okay, usually you'll get all these questions for 2 marks or 1 marks. You must keep this in your mind. Even during Viva also, this will be asked. Okay, next one is let us go through biological fluids. Okay, serum is one of the important components of animal cell culture. right? Which is the source of various amino acids, hormones, lipids, vitamins, polyamines, and salts containing ions such as calcium, ferrous, ferric, potassium, etc. Okay. It also contains the growth factors which promotes cell proliferation, cell attachment, and adhesion factors. Okay, serum is also which is obtained from human adult blood, placental cord blood, horse blood, calf blood. All these different sources of serum can be used in animal tissue culture as biological fluids. The other forms of biological fluids used are coconut water, amniotic fluid, pleural fluid, then insect hemolymph, then aqueous humor, then from eyes, that is vitreous humor, etc. Okay, all this will be used as a biological fluid during the animal cell culture. Okay, we can also prepare the plasma in the laboratory, taking out blood. Okay, and for example, here you could see we can withdraw the blood and place that in the tube. Okay, after placing in the tube, further we can go through centrifugation. Okay, after centrifugation, you'll be able to differentiate the blood into erythrocytes, where you could see 45% of whole blood, less than 1% of whole blood, which consists of leukocytes and platelets that can be seen the buffy coat below the plasma. This is the plasma, which is the straw yellow colored fluid of the blood, or liquid part of the blood is known as the plasma. Whereas uh, this is the cells. You can see here the cells, the leukocytes, platelets, erythrocytes, which comprises of this particular portion where you could see the cells. Okay, whereas this is the plasma, which is of 55% of whole blood. And these leukocytes, platelets, and erythrocytes, which are known as formed elements. Okay, further here, when you put clotting, um, when you do not put the any kind of clotting factors like heparin or anticoagulants, like when you add the anticoagulants like sodium citrate, what happens? Your blood will be in the liquid state. When you do not add the anticoagulant, okay, that time what happens? All the blood cells means formed elements like leukocytes, platelets, erythrocytes, which will be set at the bottom forming the clot. Okay, this is the clot, blood cells in fibrin clot, and this is the serum, which is minus of fibrinogen, okay, or clotting factor. And this is the plasma, which contains fibrinogen, okay. Whereas below the serum, you could see the buffy coat, where you can see the white blood cells and platelets can be seen in this particular buffy coat. And here you can see the red cells, okay. When you do not put any kind of anticoagulants here, all the cells will get coagulate with each other and you are going to get the clot formation. Okay, and then in the tube, you'll be able to find serum, which is devoid of fibrinogen. In the clot, you'll be able to find the blood cells in fibrin clot. Okay, now let us go through here tissue extracts and embryo extracts. Okay, tissue extracts, for example, are embryo extracts. Extracts from tissues such as embryo, liver, spleen, leukocytes, tumor, bone marrow, etc. are also used for culture of animal cells. Okay. Further, bovine embryo extracts are also prepared using bovine embryos of up to 10 days age. Okay, you can use this particular bovine embryo extract in the animal cell culture. Okay, further let us go through tissue extract. That is, extract some tissues such as embryo, liver, spleen, bone marrow, leukocytes, tumors, etc. are used. Right? I've already told you in this case, right? Further, we can also add this information here. Tissue extracts can often be substituted by a mixture of amino acids and other certain organic compounds. Okay. Then now let us go through advantage or disadvantage of natural media. Natural media is more beneficial to tissue culture process for high cell growth and proliferation rate. Okay, what are the limitations here? Like poor reproducibility due to lack of knowledge of exact composition of this type of media. That's the only drawback or limitations, and contamination risk can be seen in this particular uh, natural media. Okay. Now let us go through here artificial media. Artificial media contains partly or fully defined components. Okay. The basic criteria for choosing an artificial media for animal cell culture are it should provide all the required nutrients to the cell. Okay. Media should maintain the physiological pH at around 7 with the help of buffering system. The animal cell culture media should be sterile and isotonic to the culturing cells. Okay, the basis for the animal cell culture media is the balanced salt solution. In short, we can say this as BSS, which are used to create a physiological pH and osmolarity required to maintain the animal cells in vitro or in laboratory conditions. Okay, for promoting cell growth and proliferation, many types of animal cell culture media are designed by adding or varying different constituents. Example, serum containing media and serum free media. Okay. Next, let us go through here artificial media. Artificial or synthetic media are prepared by adding nutrients, that is both organic and inorganic. Okay. Then further by adding vitamins, salts, oxygen, and carbon dioxide gas phases, serum proteins, carbohydrates, cofactors. All this can be seen in this particular artificial media. Okay, let us go through here. Different artificial media have been devised to serve one or more of the following purposes. Okay, the first one, that is immediate survival. Okay, a balanced salt solution with a specific pH and osmotic pressure. Okay, then prolonged survival, a balanced salt solution supplemented with various formulations of organic compounds and our serum can be observed. Okay, then indefinite growth, specialized functions, all these characteristics can be seen. Then minimal criteria needed for choosing a medium for animal cell cultures. The medium should provide all the nutrients to the cells. Okay. And this medium should maintain the physiological pH around 7.0 with adequate buffering. The medium must be sterile and isotonic to the cells. The various artificial media developed for cell culture may be grouped into following classes. Okay. Those are serum containing. Here you can see the serum containing media, then serum free media, then chemically defined media, then protein free media. Okay. So what are those? Serum containing media. Okay. Then serum free media, then chemically defined media, then protein-free medium. Okay, before going to all this, let us understand what is plasma or serum. Plasma, which is the pale yellow liquid, okay, remaining after cells are removed from blood, normally holds blood cells in whole blood suspension, mostly water, which contains dissolved proteins, clotting factor, electrolytes, hormones, etc. Okay, so this is about the plasma. Then let us go through the serum. What is serum? Serum, which is the liquid extruded from coagulating blood, okay, widely used in the media for animal cell culture. And this is also a component that is neither blood cells nor clotting, and electrolytes, antibodies, antigens, etc. can be seen. Okay, so this is the difference between the plasma and serum. Kindly make a note. This is very important. Okay. Now let us go through serum containing media. Okay, serum consists of about 200 identified chemicals, okay, and many more unidentified chemicals. Okay, some of them are albumin, amino acids, glucose, calcium, steroids, okay, ascorbic acid, bilirubin, cholesterol, cortisol, creatinine, lactic acid, then 100 types of lipids, okay, then phospholipids, sulfates, steroids, lipases, acid phosphatase, clotting, degrading factors, glycoproteins, lipoproteins, growth factors, etc. Okay, this is all about the serum containing media. So what is the serum here? Serum, this is the liquid extruded from coagulating blood. Okay, that is nothing but the serum, a liquid part of the blood, okay, which comprises with antibodies, okay, and antigens, etc. Now let us go through here serum containing media. Okay. Further, this media in which some quantity of serum used with other defined nutrients. Okay, serum containing media carry hormones, vitamins, minerals, etc. What are the advantages of serum in culture media? That is, the serum provides binds and neutralizes toxins. Serum contains a complete set of essential growth factors, hormones, attachment, chemicals, and spreading factors, binding and transport proteins. It contains the protease inhibitors. Okay, it increases the buffering capacity and it provides trace elements. Now let us go through here the major functions of serum. Okay, in major functions, I've already listed here also the advantages. Okay, here this serum provides the basic nutrients for cells. The nutrients are present both in the solution as well as are bound to the proteins. Okay, kindly pay attention. It provides several hormones. For example, insulin, which is essential for growth of nearly all cells in culture. Okay, for example, cortisol, testosterone, prostaglandins, etc. It contains several growth factors, okay, like platelet-derived growth factor, in short PDGF, okay, platelet-derived growth factor, okay, then transforming growth factor, okay, beta, that is TGF, transforming growth factor, then epidermal growth factor, EGF, epidermal growth factor, fibroblast growth factor, in short FGF, okay, etc. I request everyone kindly make a note of all these particular growth factors. Okay, in my next video, I'll be discussing with the growth factors. These are present in concentration of microgram per liter. Okay. Okay, this is it's not microgram per liter. I'm sorry. This is microgram per ml. Okay. Serum provides proteins. For example, fibronectin, which promote attachment of cells to the substrate. Serum provides separating factors that help the cells to spread out before they can begin to divide. Okay. Further, serum provides several binding proteins. For example, albumin, which carries lipids, vitamins, hormones, etc. to cells. Okay. Transferrin carries iron in non-basic form. Serum increases the viscosity of the medium and thereby protects cells from mechanical damage. For example, shear forces during agitation of suspension cultures. Okay. Further, protease inhibitors present in the serum protect cells, especially trypsinized cells, from proteolysis. Okay. Serum also provides several minerals like Na plus, K plus, Fe2 plus, zinc, Zn2 plus, Cu2 plus, etc. Serum also acts as a buffer. Okay, this is all about the major functions of serum or advantages of serum also we can consider. Now let us move on to disadvantages of serum. Okay, serum may inhibit growth of some cell types, for example, epidermal keratinocytes. Serum may contain some cytotoxic or potentially cytotoxic constituents. For example, fetal cough serum, which contains the enzyme polyamine oxidase, okay, which converts polyamines like spermidine and spermine, which are secreted by fast-growing cells, okay, that into cytotoxic polyaminoaldehyde. Serum quality varies from batch to batch. Hence, it requires costly and time-consuming and testing every time when a new batch has to be used. This is one of the major disadvantage. Growth factors may be present in sufficient concentration. Hence, they may need external addition. Insufficient. Growth factors may be present in insufficient, okay, concentration. So when there are insufficient growth factors, that must be provided through external condition. Okay. Serum and its components interfere with downstreaming processing. So contamination with the end product is common when we use this particular serum as a natural media, and the supply is lower than the demand. Reproducibility is not possible when we use serum in the medium. Okay, and here it cannot be standardized growth conditions for cell lines when serum is used, because we do not know the exact chemical component within the serum, as this is the one of the undefined media. Okay. Next, presence of serum in the culture media may interfere with the purification and isolation of cell culture products such as pharmaceutical compounds. Okay, these are some of the disadvantages of serum. Further, to continue some of the other information, it is not chemically defined and therefore its composition varies a lot. Okay, because serum which comes under the natural medium. It is sometimes source of contamination by viruses, mycoplasma, plants, etc. The serum increases the difficulties and cost of downstream processing, where you are going for the extraction methodology, that which requires very higher amount. It is the most expensive component of the culture medium. Being highly viscous, the sera slows down the sterilization by filtration of the media. Okay. And last disadvantage here, from time to time, there is a shortage in world supply of serum. Okay. These are some of the disadvantages of serum in culture medium. Now let us go through here serum free media. Okay. Presence of serum in the media has many drawbacks and can be can lead to serious misinterpretations in immunological studies. Okay, a number of serum-free media have been developed. These media are generally specifically formulated to support the culture of a single cell type and incorporate defined quantities of purified growth factors, lipoproteins, and other proteins. These media are also referred as defined culture media. Since the components in these media are known, only this particular serum-free media will be known as defined culture media. So this defined serum-free media chemically, which is defined medium, not having complete serum, but they may but they may have some defined components of serum. Okay, investigations were made to develop serum-free medium because of the disadvantages due to the serum. The efforts were mainly based on the following approaches. Okay, one is the analytical approach, where based on the analysis of serum constituents. Okay, other one is the synthetic approach to supplement basal media by various combinations of growth factors. Limiting factors approach, like consisting of lowering the serum level in the medium till growth stops, and then supplementing the medium with vitamins, amino acids, hormones, etc. till growth resumes. These approaches have resulted in several elaborate media formulations in which serum is sought to be replaced by a mixture of amino acids, vitamins, okay, then several organic compounds, hormones, growth factors, and other proteins are supplemented when required. Okay, why are we using here the serum free media here? We are using the serum free media because of the disadvantages which is caused due to the natural media like serum media. Okay, and this serum free media, which can also be known as the defined medium. However, addition of 5 to 20% of serum, even in these media, is essential for optimum growth. Okay, like MCDB 202, chick embryo fibroblast, CMRL 1066, continuous cell line, MCDB 110 202, fibroblast, human diploid fibroblast, NCDB 402, fibroblast, mouse embryo fibroblast, 3 T3 cells, MCDB 130, skeletal muscles, HOS, mouse leukemia, mouse erythroleukemia, the skeletal muscles. Okay. Now let us go through here advantages of serum-free media. By using this serum-free media, reproducibility results is possible. And here, this is easier downstreaming or processing. Hence, cost can be reduced. Toxic effects can be completely reduced. Then, bioassays are free from interference due to the serum proteins. No danger of degradation of sensitive proteins. allow selective differentiation and production of specific cell types from heterogeneous cell culture. Okay. In serum-free media, end product can be recovered without much downstream steps. Then this serum-free media also will lead to the drug discovery, physiological expression, and gene expression study, which has become simpler to perform. Okay, these are some of the disadvantages of, advantages of serum-free media. Now let us go through here further again advantages of serum-free cell culture. Low risk with regard to contamination with the bacteria, fungi, and viruses. Okay, and it is better defined and reproducible formulations allow more convincing and comparable research results. Time consuming where batch tests are not necessary. Here you can see in serum-free media the elimination of source for possible infections, infectious agents, for example, viruses, prawns, etc. Fulfillment of legal conditions for the production of medical products, reduction of contaminations of the end products by the culture residues. Okay, now let us go through the disadvantages. Slow cell proliferation, okay, whereas in this case, most of the serum-free media are not as efficient as serum-added media in the growth promotion of cells. What are we doing here now? We are going through disadvantages of what? Of serum-free cell culture, okay, or serum-free media. This need for multiple media. This needed for multiple media, that is a large number of serum-free media need to be developed for different cell lines. Then purity of reagents. Here what happens, the native serum does possess some amount of cleansing effect on the apparatus and reagents. And therefore, in the absence of serum, pure grade reagents and completely sterile apparatus should be used. Okay, hope you understood the further. Some of the other disadvantages of serum-free media are generally it is specific to one cell type. Many media required for different cell types. Then this is time consuming for preparation of specific ingredients is more, and greater care is needed in control of pH, temperature, osmolarity, and carbon dioxide concentration. Growth rate and cell density is very low in the serum-free media. Okay. Lastly, cells become more fragile in long-term cultures unless biopolymers or synthetic polymers are used. So this is all about the disadvantages of serum-free media. Okay, from the beginning, what are we went through? This unit is very important. Kindly pay attention. Okay, in this unit, you are going to get three different types of media. Okay, you may get the questions like what are culture medium or culture media and give their types, and about the normal cells or immortalized cells or transformed cells are basic requirements of the culture media. Okay, then about the natural media here, then clots, you may get this one for 2 marks or 1 mark, or coagulant form also you may get in the examination, or biological fluids for 2 marks you may get this biological fluids. Okay, then you may get this serum free media. All these are very important: serum containing media, serum free media, chemically defined media, and protein free media. All these are very important. You must keep in your mind. Even the plasma, then serum definition. Okay, serum containing media, 2 marks or 5 marks usually then ask. You must be able to write. When they asked about the serum containing media, define what is serum, then explain about the serum containing media. Okay, and the major functions of serum, advantages part you need to mention here. Then you can also mention about the disadvantages of serum. Okay, Further, this is about the serum free media. You must make a note. Okay. Then the efforts were mainly based on the following approaches. All those you can keep in your mind. And also, you must keep in your mind about the, some of the essential media which are required during the growth of the some of the cells. Then here you have given advantages of serum free media. Okay, then disadvantages of serum free media. Okay, so this is all about the culture medium. In this, I have focused more on natural media. Kindly go through that one. I, Dr. Pratibha K, signing off for today. Stay tuned with me. I'll be back with some more videos. Thank you for watching.